In this video, we are going to be learning about enzymes and how their structure dictates function. Before we can learn about enzymes, we need to understand chemical reactions, because enzymes speed up chemical reactions. When we focus on chemical reactions, we're going to look at the energy involved. And one of the types of energy we're going to pay attention to is called free energy, also known as delta G or Gibbs free energy. Now, free energy is a reflection of the ability of a cell to do work. Now, there are two different types of energy-related reactions we're going to look at: exergonic reactions and endergonic reactions. Let's start off with exergonic. Exergonic have a negative delta G. What that means is they have a net release of free energy from their environment. These are often catabolic processes. That means that they are digestion reactions, or things like hydrolysis, or something like cellular respiration, which is a multi-step process where the cell breaks down food to release energy. So let's look at this graph of cellular respiration. So our y-axis demonstrates the amount of potential energy. So that is the energy that has the potential to do work. So that's basically, think of it like stored energy. So the reactants have a lot of potential energy, mainly because of glucose. And as we learned about the structure of glucose, we know there are many carbon and hydrogen and oxygens bound together, and all of those bonds are storing energy. Now, as glucose and oxygen go through the catabolic process of cellular respiration to produce the products of carbon dioxide and water, we can see that the products have much lower potential energy. Now, the difference between the energy in the reactants and the energy in products, this is the delta G. That's the energy output. And since it is a decrease, we have a negative delta G in this case. We're releasing energy to the environment. On the other hand, a positive delta G represents an endergonic reaction. Ender, think enter. Okay, there is a net input of free energy from the environment. These are building reactions, so anabolic processes, things like synthesis, dehydration synthesis, or in this case, photosynthesis. Photosynthesis is essentially the reverse of the chemical equation for cellular respiration. Okay, in this case, this plant cells are going to take the low energy reactants, okay, of carbon dioxide and water, and they're going to produce high energy products of glucose and oxygen. Now, that means we're going from low energy reactants to high energy products. That means we have to put energy in, and in the form of photo or light. So light energy is helping power this process. So in this case, because we're putting energy in, this is going to be a positive delta G. All right, another form of energy we need to pay attention to in a chemical reaction is the activation energy. Activation energy is the amount of energy the reactants need to obtain in order to carry out a chemical reaction. We abbreviate this as EA, okay? So let's take a look at this top graph first. Okay, we have our reactants and we have our products. Okay, so notice our reactants are going to have different of our colored elements bonded together than our products do. Okay, and so over the course of this reaction, bonds are going to break and new bonds are going to form. However, the reactants are currently stable. Okay, they are making the correct probably covalent bonds, and so energy is needed to be added to get those bonds to begin to want to break so that new bonds can reform. Now, this little input of energy to kind of get the ball rolling, okay, to get the molecules in this kind of unstable transitional state, that is called the activation energy. Okay. Now, the activation energy, okay, is going to be the difference between the reactants and this kind of peak of potential energy. Okay, so or free energy. So that is that input needed to get the reactants to kind of become unstable so that bonds can break and we can form the products. The delta G does not change. The delta G is still going to be the difference between the reactants and the products. So in this case, this is an exergonic reaction, okay? And so we're gonna have a negative delta G, which is what we can see here in this graph. So one more time, delta G is still going to be the difference between the reactants and products. However, in order to destabilize the reactants so they become the products, there needs to be an input of energy, and that hump is the activation energy. Now, for an endergonic reaction, again, delta G, difference between reactants and products. In this case, we have a positive delta G we have to put in. But in order to get the reactants destabilized enough to form the products, we have a very big hump. Okay, so remember the activation energy is the difference between the reactants and that peak free energy. So here we have a large activation energy. A lot of energy has to go in to get an endergonic reaction going. It is what we would say not spontaneous. You have to put a lot of energy in. Exergonic reactions are spontaneous. However, that does not necessarily mean they're fast. Okay? So for example, glucose will eventually break down on its own to form carbon dioxide, or starch will eventually break down into individual glucose molecules. That can happen on its own. However, it will take a very long time because you have to get this activation energy in order to have that reaction occur. So chemical reactions can happen, but they're very slow processes, and that's where enzymes come into play. Enzymes are proteins that catalyze chemical reactions. So that means they speed up chemical reactions. And they do this by reducing the amount of activation energy needed for a reaction to occur. So let's take a look at our exergonic and our endergonic reactions one more time. So this is our exergonic over here on the left. You'll notice my energy of my reactants and the energy of the products are staying exactly the same in both of these lines. Okay, so delta G is unchanged. However, what is changed is that size of that activation energy hump. With a red arrow, okay, or our red line, sorry, the activation energy for an enzyme is going to be much slower or much lower than the activation energy without an enzyme. That means with an enzyme, less energy needs to be absorbed from the environment to get the process going. And because less energy is needed, the reaction can in general happen faster. With an endergonic reaction, we have really large differences in activation energy. We've got big activation energies, right? Because we have low energy reactants to get to high energy products. So without an enzyme, this black line, you can see that the hump is very large. But the red line, this activation energy with an enzyme, it's lower. So there's still a large energy input, it's just less. So once again, we see enzymes decreasing the amount of activation energy needed. So how exactly do enzymes lower activation energy? Well, it's all about their structure. So let's start off by talking about the fact that enzymes are proteins. And as we learned before, proteins have unique 3D shapes. Because that polypeptide folds and it goes from primary to secondary to tertiary, okay, it's going to have a unique 3D shape. Sometimes proteins will have a quaternary structure as well. So all enzymes, as they fold, the polypeptide folds, they are creating these kind of nooks. And the main nook in an enzyme is called the active site, okay? And so that is this area right here, okay, that I'm highlighting, okay? So an active site, right there, an active site is where the substrate binds. And substrates are molecules that are acted on by enzymes, okay? So you can see this kind of tealy blue substrate fits into the active site. Okay, now because they are also 3D shapes, they might have some additional grooves. The grooves away from the active site in an enzyme are called allosteric sites, and that is where regulatory molecules can bind, such as this inhibitor we see down here. So I'm going to highlight the allosteric sites, green in this case, so you can see they're away from it. You'll learn more about allosteric sites and regulatory molecules in the next video. So the main thing you need to remember is that enzymes have a unique active site which fits perfectly with its substrate. So because of that, enzymes are specific. Because of its active site shape, each enzyme can fit with a specific substrate. So here, this is an example we've talked about previously. Okay, you'll see that we have starch, which is a 1,4 linked glucose. Okay, and you can see that amylase, which is the enzyme that digests starch, the active site fits perfectly with that specific bond, that glycosidic linkage between our sugars, between glucoses. But with cellulose, okay, we have beta 1,4 linkages. So you can see that in some cases, the bonds are kind of different shaped. They're pointing up rather than down. And amylase cannot fit with that correct bond, and so it can't break it down. So that is why, one of the reasons why our body cannot digest cellulose well, but it is able to digest starch. So we often call this idea of substrates and active sites fitting together like puzzle pieces, like the lock and key model. So you can see down here, here's the substrate. It's going to fit into this active site of the enzyme just perfectly because they have a matching shape. Kind of like a key fits into a lock, as you can see here. Okay. But this doesn't give the full explanation of how exactly enzymes reduce activation energy then. So the reason why they reduce activation energy has to do with something called induced fit. So the binding of the substrate on the enzyme's active site causes the enzyme to undergo something called a conformational change. And what that does is it leads to a tighter fit. Okay, a conformational change is a shape change. So usually it's the binding induces a shape change, hence the name induced fit. And in this case, the binding of the substrate induces the active site to fit more tightly around the substrate. Okay, I like to think of it as like a squeezing. Okay, so the enzyme is going to squeeze the substrate, either squeezing and putting strain on existing bonds, in the case of digestion. So you can see here, here's our enzyme with our active site. You can see when the substrate binds, you can see that the enzyme almost begins to wrap around that substrate. It's squeezing it, okay? And so it's squeezing existing bonds so that products can be made. Or if we have a synthesis reaction, so here are our two substrates, okay? They're going to fit into the active site. And because in this case, when we're synthesizing, the enzyme is going to hold and orient the substrates just right so that bonds are easier to occur. So this kind of, again, this squeezing in this case is almost bringing the substrates together at the right angle for a reaction to occur. So let's just look at the general operation of enzymes first and talk about some important ideas. So let's work around this kind of cycle. Okay, so our first step one, okay, the enzyme is available with an empty active site. In this case, our enzyme is called sucrase. Okay, I want to point out that many, not all, but many enzymes end in ase. That's a good way to tell what type of molecule you're dealing with. So sucrase's substrate in this case is the sugar sucrose. Ose is usually the ending of a sugar or carbohydrates. So in this case, the substrate sucrose has the right shape to fit into the enzyme sucrase. Now, we can see that there is an induced fit occurring. We can see this active site is kind of wrapping around the substrate, okay? And when the active site kind of wraps around the substrate, we call this the enzyme substrate complex. They are bound together in this kind of induced fit model. Now, if this enzyme is going to break this substrate down and digest it, then we need to have hydrolysis. So we have to add water to break that bond, okay? And in this case, the substrate is going to get converted into two separate products, which in this case are glucose and fructose. The products get released, and it's really important to notice that once the products leave, we go back to the enzyme as it was before. So an important idea is that an enzyme is unchanged in a reaction, and therefore it can be used many times over and over again. So let's just look at some more examples just to understand why induced fit actually reduces the activation energy. So let's look at this digestion example. This enzyme is a type of dipeptidase. So in this case, it is digesting a dipeptide into 2 amino acids. Okay, now you can see that this dipeptide is coming into the active site of the enzyme, and because of induced fit, the enzyme is essentially wrapping around those 2 amino acids and putting stress on the peptide bond between them. And that is then allowing for these 2 separate amino acids to then be released from the enzyme. The enzyme is unchanged. Okay, so in digestion, the enzyme's induced fit squeezes existing bonds, allowing them to break easier, puts stress on them. In a synthesis reaction, the active site is going to orient the substrates and bring chemical groups closer together so that they can carry out a reaction. So in this case, we have the enzyme maltose synthase. Okay, it's going to synthesize maltose, which is a disaccharide of 2 glucoses. So here you can see we have 2 separate glucoses, okay, and in the process of dehydration synthesis, okay, water is going to be removed, okay, and as a result, we're going to get our product, which is this right here, which is our disaccharide maltose, okay. So in the case of a synthesis reaction, it's speeding it up because these 2 glucoses have their hydroxyl groups oriented towards each other so that dehydration synthesis can happen faster. On their own, these glucose molecules would have to be heated up a lot to gain enough energy to bump into each other and orient so their hydroxyl groups are just right to do dehydration synthesis. And it would take a lot of energy to do that. Versus the enzymes, because of the active site shape, orients the molecules so that they're ready to form those bonds needed. So let's just summarize enzymes. Enzymes are proteins that lower the activation energy of reactions. They are reaction specific due to their shape of their active sites, right? Like a lock and key. Every substrate fits into the active site of a specific enzyme. Okay, we have the specificity. They are reusable, so they are not consumed in the reaction. The enzymes are unchanged. And then our final point is that they're affected by cellular conditions because they're proteins. And that is what you're going to learn about in your next video.